Hello everyone, today we are going to discuss about the chemical spectroscopy infrared spectroscopy. Now we are going to introduce ourselves. My name is Tan Chuan and my role is leader and editor.
My name is Bernard Lim Zije and my role is as a researcher and presenter.
My name is Kitchen and my role is as a researcher and a presenter.
My name is Menaka Rajamani. My role is editor and researcher.
My name is Dehuizi and my role is researcher and presenter.
My name is Yuzian and my role is researcher and presenter.
So, what is an IR? Now let me introduce you. IR is a measurement of the interaction of infrared radiation with matter by absorption, emission, or reflection. It's used to identify chemical substances or functional groups in solid, liquid, or gas. It can be used to verify known and unknown samples. The technique of IR is conducted with an instrument called an infrared spectrometer, which produces an infrared spectrum. An IR spectrum can be visualized in a graph. This is the example of the spectrum on the slide.
Now let's have an overview of the technique, which includes principle, instrumentation, and application. When IR radiation interacts with a sample molecule, the sample molecule will absorb the specific applied contact IR light, which is matched with the internal vibrational frequency of the molecule, and other residual frequencies are transmitted via the sample. Due to absorption of IR radiation, the next change of dipole moment in the molecule occurs and causes vibration of bond in the molecule, like stretching and bending vibration. The transmitted light is detected by the detector and IR spectrum interpreted on the computer screen by analysis of the transmitted light. The band corresponds to the characteristic functional group and the bond present in the chemical substance.
IR spectroscopy measurement is around 2.5 to 25 micrometer. The IR spectrum is typically divided into three regions, which are near IR, mid IR, and far IR. Molecules absorb specific wavelength of IR radiation. This causes their bond to vibrate. There are two types of molecular vibration, which are stretching and bending. Examples for stretching vibration are symmetrical and asymmetric. Next, bending vibration can be differentiated to two types, which are in plane and out of plane. Scissoring and rocking are in plane bending, whereas wagging and twisting are out of plane vibration. Next, to interpret or analyze IR spectra, it involves functional group region and fingerprint region. For functional group region, spectrum peaks in this region are associated with specific functional group like OH, NH, CH, CO, and others. This is the diagram showing typical infrared absorption values for various types of bond.
The main part of IR spectrometer includes a source of radiant energy which emits IR radiation. Second is monochromato. Third component is sample cells and sampling of substances. Fourth is detector, and lastly recorder are used to record the IR spectrum. There are several applications of IR spectroscopy, including chemical identification, compound purity testing, and quantitative analysis.
Now I'm going to share with you all about the highlight of interviews with the IR lab assistant in UPM. Hai semua, hari ini saya akan interview dua IR lab assistant dalam UPM. So, boleh introduce yourself, Kak?
Saya Nurhana Miza binti Nuri, bekerja sebagai PIC FDR selama 7 tahun selepas dipindahkan dari biotech. Saya Syakir Azmi, saya sektor PIC untuk alat ICIR. Saya dah bekerja sejak 2019 dan sekarang dalam 5 tahun dah.
I see. What are the roles that research has during everyday working as an IR lab assistant?
Untuk FDIR, kami akan menerima sampel student, menguruskan pembayaran dan perang sampel lah.
Okay.
Sama juga lah macam yang saya katakan, semuanya sebab kita ada satu instrumen, saya akan rutin lah.
Well, it seems quite difficult and challenging. So, research, have you faced any challenges in this field so far from when you first step into this industry?
Challenges we face, first, untuk pastikan keadaan bilik ini, humidity kena very low. Ya, very low. Persekitaran juga yang kami hadapi sekarang, macam bangunan ni terlalu butuh. So, kita ada kita pernah menghadapi kebocoran air. So, itulah antara yang kami hadapi sekarang. So, bila menghadapi kebocoran, humidity menjadi tinggi, dia menyebabkan alat menjadi rosak. Bila rosak, kos yang diperlukan amat tinggi untuk FTIR.
So, Mrs. how do you overcome them by using the knowledge that you learn from the past?
Biasanya untuk pastikan humidity tak kawan humidity, first aircon operate 24 jam. So sebab sekarang 3 aircon ni akan ikut timer, kami set timer. So dia akan berjalan lah setiap 24 jam.
So, Mrs. can you share with us about what you have gained from your expertise throughout this year in a few weeks?
Yang saya cakap sebelum ni, saya dalam background chemistry. Sebelum ni saya cuma tahu macam mana nak sampel je. Apa tak ada sampel. Jadi sekarang bila dah masuk kerja ni, kena tahu semua benda. Dari segi dia punya maintenance, macam mana nak tukar speaker semua, gas mana nak tukar, macam mana nak tukar gas. Itulah.
Next, I'm going to share some practical impacts of IR spectroscopy in bioscience field, which include biomedical application and in the forensic science field. First, IR spectroscopy is used in the bioanalytical application. It is due to the non-destructive, labor-free, and cost-effective way in acquisition of biochemical information. So IR spectroscopy now is widely used in the examinations of many of cells, tissues, and organs. An example is the examination of study bar for healthy persons and COVID-19 patients. In clinical application, diagnosis of carcinoma is possible as IR spectroscopy demonstrates high potential to show the carcinoma symptoms. Let's look at an example. The first spectrum shows differences for healthy people and the carcinoma patients. Another one, which is the second spectrum, is for the healthy people and for those with breast cancer, as well as for the third one, showing the differences between the healthy person and the lung cancer patient. IR also can use to detect the urine disease outcome by analyzing the body fluid. Some differences can be observed in the spectrum, like as shown in the slide. Next, IR spectroscopy can be applied in the forensic science field. It's useful in the crime scene to detect and differentiate different compounds like the saliva, sweat, and the blood, especially for blood, which is the most informative but is most difficult in visual differentiating. And also, this method is not destructive towards the crime scenes. For example, the spectrum here differentiates the blood of different organisms, and also we can know the edge of the blood stain, how long the blood has appeared. Another example here, the first spectrum is to differentiate between the different body fluids like the blood, saliva, semen, urine, and sweat. And also the second spectrum tell us what is the postmortem blood and the antemortem blood.
Now I will explain about the IR spectrometer instrumentation using schematic diagram. Basically, there are two types of spectrometers, which are dispersive and Fourier transform spectrometer, FTIR, where it is the latest instrument. Here is the schematic diagram of FTIR, consisting of source, interferometer, sample compartment, detector, amplifier, AD converter, and a computer. Thus, the source generates radiation and passes the sample through interferometer and reaches the detector. Then the signal is amplified and converted to digital signal by amplifier and analog to digital converter to the computer. Finally, data is produced in the form of IR spectrum. Next, let's watch a short video on the demonstration of FTIR.
Prepare the sample by crushing it into powder. Clean the instrument with acetone, then control the settings using Opus software. Then place the sample in the holder and scan the sample. Finally, the IR spectrum is obtained. Don't forget to clean the instrument before the next use.
I hope you all have a clear idea on how FTIR works. In conclusion, IR is a powerful tool for identifying molecule structure by analyzing how bond absorb infrared light. That's all from us. Thank you.